Hello, good evening everyone. How are you all doing? In this video, we'll be talking about the biology of a coccidian parasite named Toxoplasma gondii. And in the previous video, we've talked about the Plasmodium species, and we mentioned that Plasmodium species are also sporozoans, and they follow a typical life cycle of an apicomplexan. And we said the reason why they are called apicomplexan is because they possess a particular structure, a structure known as apical complex. So, Toxoplasma gondii is also a coccidian parasite, just like the Plasmodium, and it follows a what? It follows a typical apicomplexan life cycle. But today we'll be saying more about Toxoplasma gondii. What is Toxoplasma gondii itself? Toxoplasma gondii is a tissue cyst forming coccidian parasite, and it infects a wide range of hosts. But it is primarily a parasite of the felids. Primarily a parasite of the what? Of the felids. So, when you talk about felids, you mean what? You mean cats. So they are what? They are primarily parasites of what? Cats. But they can also have some other intermediate hosts, such as swine, rats, pigs, and even man himself. So a man can also be what? A um, an intermediate host. You understand? Now, they cause a disease known as toxoplasmosis. Toxoplasmosis, and let me show you a slide. Toxoplasma gondii is an obligate intracellular coccidian parasite that was firstly described by Nicole and Mankios in a small American rodent known as called Gondii. So that is where they saw the generic name. What? The generic name Gondii. You understand? Now, it is an important pathogen that was firstly observed in 1923 in the retina of a child. In the retina of a child with hydrocephalus and microphthalmia. The name Toxoplasma is derived from a Greek word, toxon. Toxon means arc, like an arc, or a brow, referring to the curved shape of the what? The curved shape of the trophocyte. So, the name toxon was gotten from a Greek word, that is toxon, which means what? Which means an arc. So an arc is what? Something that is curved. You understand? So let me show you the image of the trophocytes. So look at the trophocytes. If you notice very well, the trophocyte look like what? Like this. It is curved. You understand? So that is where they get the image. That's where they get the name of what? The name Toxoplasma. And Gondii is from the what? From the rats that it was first observed, which is what? Which is named Gondii. So the name what? Toxoplasma Gondii. Now, what are the things that we are going to what the thing that I want you to notice in this particular um, trophocyte structure is just this: the presence of what? The presence of apical complex. Hence, they are what? They are apicomplexan. Apicomplexan. You understand? They are what? Apicomplexan, because they possess what? They possess an apical complex. You understand? So these are other structures that they possess apart from apical complex, the micronames apicoplast, the centrosome, Golgi body, dense granules, endoplasmic reticulum, mitochondrion, and basal complex, and other stuff like that. So now let us proceed with what you're saying. Toxoplasma gondii is now recognized as one of the most protozoan parasites that affects wide range of hosts, and I've mentioned that they are what? They are felids parasites. So the definitive host is what? Is felid. It's cats. You understand? It's cats. You understand? Now, they now exist in three forms. The first form is the trophocytes, the second form is the tissue cyst, and the third form is the oocyst. You understand? The trophocytes, the tissue cyst, and the oocyst. The trophocyte and the tissue cyst represent a stage in asexual multiplication, asexual multiplication, while the oocyst is formed by sexual reproduction, which can be gametogony or sporogony. You understand? So the trophozoites and the tissue cysts are formed by what? By schizogony. And the oocyst is formed by gametogony or sporogony. Now, don't forget what you have mentioned before when we were talking about the life cycle of a typical apicomplexan. We mentioned that a typical apicomplexan has a 3-stage in their life cycle. We mentioned the 3. We said we have the schizogony, which is an asexual life cycle. We have the gametogony, which is asexual, and we have the what? We have the sporogony, which is asexual. So I said they have what? They have sexual, asexual, sexual life cycle. Asexual, sexual, asexual life cycle. Right? Now, that one is gone. I've described what a Toxoplasma gondii is. I've showed the structure. This is the structure of Toxoplasma gondii under the microscope. Curved or arc shape. Can you see? It is curved. It is curved. With a what? With an apical complex on the what? On the top. And they use apical complex for what? For attachments. For attachments to their host. You understand? And I've already mentioned that they exist in 3 forms. The forms is what? The trophozoite. Right? The trophozoite, the tissue cyst, and the oocyst. So these are the 3 forms that they exist. Now this tissue cyst can now be 2 as well. We'll talk about it. That is either the bradyzoites or the tachyzoites. We'll talk about it. Now let's talk about the life cycle of the Toxoplasma gondii. I've mentioned the forms of Toxoplasma gondii, and I'm mentioning that all the 3 forms exist in what? In cats and other felids, which are definitive hosts and support both schizogony and gametogonic life cycle. It should be noted that when I said the word felids, I did not mean only domestic cats. No, I mean all the cats, including the big cats like lions, tigers. You understand? Now, only the asexual forms, trophozoites and tissue cysts, are present in other animals. You understand? Including humans, birds, pigs, swine, which are intermediate hosts. You understand? Now, all the 3 forms are infectious to man. Now, remember I mentioned that we have the trophozoites, the tissue cyst, and what, and the oocyst. They now have heterogeneous life cycle. We're talking about the life cycle. They have heterogeneous life cycle. When you say heterogeneous life cycle, it means they have what? They have both the definitive host, and the intermediate host. But one thing about it is that the intermediate host is not really necessary. And I will tell you the reason why it is not necessary. They are not really obligatory in the life cycle of Toxoplasma gondii. Either they are present or they are not present. Once the definitive host still exists, they are still going to be what? They are still going to continue to exist. But it should be noted that all infection are either by oral fecal routes, transplacenta, or by carnivorism. I said what? I said oral fecal, transplacenta, or what? Or carnivorism. The life cycle now involve two types. They have 2 types of life cycle, or 2 distinct phases. We have the enteric life cycle and exoenteric life cycle. We have the what? We have the enteric and the exoenteric life cycle. Also known as intestinal life cycle and extraintestinal life cycle. Intraintestinal life cycle. Some textbooks call it enteroepithelial life cycle and extraepithelial life cycle. All the things saying that if you're talking about enteron, you're talking about digestive system. You understand? digestive system. And if you're talking about digestive system, you're talking about what? You're talking about guts. You understand? So when you hear the word enteric life cycle, you're talking about guts. So if you're talking about enteric life cycle, it means the one that happens inside the guts, either the intestine. You understand? If you're talking about exoenteric life cycle, just like exoerythrocytic life cycle, outside the red blood cell, just like this one now. Exoenteric life cycle, it means outside the guts, the life cycle that exists outside the guts. So what we have mentioned here is that they have what? They have the enteric and what? The exoenteric life cycle. So let us talk about the enteric life cycle first. You understand? Enteric life cycle occurs only in the definitive hosts. It cannot occur in the intermediate host. So let me just represent this with the guts of a felid host. Remember that digestive starts from the mouth. So let me say from the esophagus, we have this. Let me use it like this. The slide is not that big. So let me just say so this is the guts. This is the guts of a felid host. You understand? Now, an oocyst. The life cycle begins when a viable oocyst is taken in by the what? By the definitive host. A viable oocyst is taken by a definitive host through contaminated food or water. Now, how does their oocyst look like? Their oocyst is like this, and the oocyst have what? It has sporocysts, 2 sporocysts. Each sporocyst now have 4 sporozoids. Understand? So this is the oocyst. Oocyst. This is the sporocyst. The sporocyst. And inside the sporocyst we have the what? We have the sporozoids. You understand? Now, after taking in a viable oocyst, once it enters into the guts of the definitive host, it ruptures to release the sporozoites that is inside it. You know, it is oocyst covered by sporocyst covered by sporozoites. But the infective, what we need is the sporozoites. You understand? So it ruptures to release the sporozoid that is inside. So we are going to have the sporozoid inside. The sporozoids, once it enters into the gut, it develops into a what? Into a trophozoite. That is what? That is like this. You understand? The trophozoite now feeds. You understand? It feeds. Then it matures into a what? Into a scission. Just like the plasmodium parasites. The scissions undergo schizogony. Develop several what? Develop several merozoites inside. You understand? We have several merozoites inside. The merozoid bursts, the schizoid bursts to release what? To release the merozoid that is inside. You understand? It releases what? It releases the merozoid that is inside. Are you getting me now? Once it releases the merozoid that is inside, the merozoid reinvade other cells. You understand? It reinvade the cell of the what? The cell of the guts. You understand? Then becomes a trophozoite again. You understand? Become a trophozoite. From a trophozoite to schisons. From schisons, undergo schizogony to give what? To give merozoites. You understand? It undergo another schizogony again. Become trophozoite. From trophozoite to schisons. Schisons releases merozoites. They reinvade again. So they continue like that. After several schizogonic life cycle, some merozoites will now decide that we want to differentiate. They are going to differentiate themselves inside the gut, still inside the gut. They are going to differentiate themselves into what? Into the male and the what? And the female gametocytes. You understand? They are going to differentiate themselves into male of what? And female gametocytes. Remember how the male and the female looks like. The male is what? We have a flagellum. You understand? We have a flagellum. Then the female will be what? Will be immotile. It won't be motile. The male will fertilize the female gametocytes. You understand? Then they give rise to what? They give rise to a motile ookinete. Ookinete now penetrates still in the guts, not outside of the guts. They are still in the guts. So it undergoes sporogony. It undergoes what? Sporogony. It undergoes sporogony and develop from ookinete to oocysts. From what? From ookinete to oocyst. You understand? The oocyst will now be taken into the large intestine. Then, oocyst is just like a cyst. It forms a what? It forms a cyst wall. It forms a cyst wall. You understand? This is the cyst wall. Space is the cyst wall to protect the what? To protect the what? The sporozoites and the sporocyst that is inside. So, taken to the large intestine, then to the rectum, then it is passed out through the what? Through the fishes. Inside the what? Inside the water. Through the fishes into the what? Into the water. Then it stays there. Then the life cycle can now continue if another definitive host now injects the viable oocyst. Then the life cycle goes like that. That is intestinal or enteric life cycle. You understand? That's the first one. So I've talked about how it goes. It still follows the typical life cycle of a sporozoan or anapi complexan. Now, that is why I said it is by what? It is by fecal oral routes from the fishes to the mouth. Now, I now said it can be what? It can be by carnivorism as well. I will explain how it can be by carnivorism. So let us explain the extra epithelial life cycle, or should I call it the exoenteric life cycle. Let us explain the exoenteric life cycle or extraintestinal life cycle. You understand? So, in this exoenteric life cycle, we are going to have it can occur in both in both definitive host and intermediate host. So, definitive host is always felids, that's cat, and the intermediate host can be man, pig, swine, rodents, and stuff like that. So we are still going to follow the same stuff. So intestinal life cycle, the oocyst, the viable oocyst is taken in. You understand? The viable oocyst is taken in. Once it enters into the gut, it develops into what? It develops into. We are going to have a release of what? The release of sporocyst, the sporozoid rather. The sporozoid matures into what? Into the trophocytes. You understand? It's going to mature into the trophocytes. Now, remember that in the intestinal life cycle, what the trophocyte is going to do is to what? Is to mature into scissions. You understand? It matures into what? Into scissions. Once it matures into scissions, becomes and goes like that, undergoes schizogony and stuff. So it goes like that, like that. But in this case, it is different. What is going to happen is that the trophocyte is going to penetrate the gut wall. You understand? It will what? It will penetrate the gut wall, go outside of the gut, and then it will be engulfed by the macrophages. It will be what? It will be engulfed by the macrophages. Once it is engulfed by the macrophages, it transforms. It transforms into what? It transforms into a rapidly dividing tachyzoid. So it's going to transform into a what? Into a tachyzoid. And tachyzoid is rapidly dividing. It is very fast dividing. You understand? Very, very fast. So it's going to transform into a tachyzoid. The tachyzoid now disseminates. It disseminates to reinvade other cell. It can reinvade the cell of the brain. It can invade the brain, the heart, the lungs. Are you getting me? The lungs, the liver, through the blood and lymphatic circulation. That is what they use for what? For circulation. So they invade other cells. They are fast dividing. They will be dividing very, very fast. But immune system is now triggered at some point. Once they reach a particular point, the immune system will be triggered. Once the immune system triggered, the immune system, it tends to slow them down because they are dividing too rapidly. They will be slowed down. Then they will now transform from a fast dividing into a slow dividing trophocyte known as the bradyzoites. And this is what we mean by the tissue cysts. So the tissue cysts is the what? Is the brad. So they stay there, slow dividing. They don't divide that much. Even at some point they are cysts. They don't divide at all. You understand? So they become a bradyzoids. I get me now. So these bradyzoids, they are cysts. They are sometimes called pseudocysts. You understand? They remain latent in the host for years, and they can become, or they can be reactivated depending on the host immunological system. You understand? The immunological system or the immunological state of the host, they can be reactivated. They stay there as a what? As a tissue cyst. You understand? So all this one, they occur outside of the intestine. That's why they call it exoenteric life cycle. You understand? Now, this exoenteric life cycle can occur in both the what? In both the felix, which is the native host, and the intermediate host. You understand? Now, these bradyzoids can now be ingested. They can be ingested by what? Through carnivorism. You understand? Through carnivorism. Maybe a lion sees a pig, or a cat sees a rat, and the rat has been infected. The rat has a bradyzoid. Then, while feeding on them through carnivorism, they are going to ingest the bradyzoid. So, a cat, which is a felix parasite, can get it from the what? Can get it from the intermediate host through carnivorism, and can get it from what? From a water through what? Through oral fecal routes. You understand? So this one, they can be exoenteric life cycle, where it goes outside of what? To form a tachyzoid, then to what? To form a bradyzoid. And this is the life cycle of what? This is the life cycle of a typical. This is the life cycle of a Toxoplasma gondii. This is what they are saying here as well. You understand? Look at it. We have the oral fecal cyst that is being contaminated food or water. You understand? Which is being ingested by what? By human. You understand? Then this one too. They pass it through each other. You understand? Now look at it. A tissue cyst, which is what? Which is a bradyzoid, which can what? Which can go into the brain, the eye, the skeletal muscles, the myocardium, which is the heart. You understand, like that. So this is just a life cycle of a Toxoplasma gondii. Now let's also go to the pathology. Pathogenicity and the clinical features. The outcome of Toxoplasma gondii depends on the immune status of the infected person. You understand? Active progression is more likely in immunocompromised individual. An example of immunocompromised individual is someone that is having acquired immunodeficiency syndrome, which is AIDS. But most infection in human are asymptomatic, except if the person is what? It is immuno compressed individual. Now, clinical toxoplasmosis may be congenital or acquired. Congenital as in what? Congenital has been transmitted transparentally from the what? From the mother to the fetus, and acquired postnatally. And when we say acquired postnatally, maybe through oral fecal routes or through blood transfusion. You understand? It can also be blood transfusion. But the most common manifestation of the acute toxoplasmosis is lymphadenopathy. That is inflammation of the lymph node. You understand? Inflammation. So the cervical lymph can be mostly affected. Fever. These are symptoms. Fever, headache, myalgia, splenomegaly are often present, and the illness may resemble mild flu, and it is self-limited, although the lymphadenopathy may persist. You understand? Now, we can have toxoplasmosis, ocular toxoplasmosis, which can affect the eye. In fact, it can cause blindness. You understand? It can cause blindness in some It can cause brain damage, it can cause paralysis as well as blindness. You understand? Now, toxoplasmosis is mostly serious when it is present in a immunocompressed individual. An example of immunocompressed individual is someone that's having AIDS. In this presence, involvement of the brain is most common. Involvement of the brain is most common, and clinical manifestation includes the encephalitis, altered mental state, seizure, cerebral sign, meningitis, and other clinical manifestation. You understand? So let's talk about the control. The control, it is very difficult to control toxoplasmosis because of wide range of animal reservoir. But currently, there is no effective vaccine available for human. A genetically engineered vaccine is under development for the use in cats, which is the what? Which is the terminative host. Now, I mentioned that the intermediate host is not really necessary in the first place. Now, the reason why I say intermediate host is not necessary is because what is formed in the terminative host is the bradyzoites and the tachyzoites, and it can also be formed inside the what? Inside the intermediate host. So, as long as the definitive host is present, Toxoplasma gondii will continue to what? Will continue to thrive. You understand? And B is the biology of Toxoplasma gondii. Thank you for listening and follow for more.